Hello everyone. The topic for today's session is histology of eye. So in this session we will learn about the histology of cornea, retina, sclerocorneal junction, the optic nerve and the eyelid. So first, the histology of cornea. It is made up of 5 layers. Those are the corneal epithelium, Bowman's membrane, corneal stroma, Descemet's membrane and the endothelium. Corneal epithelium, the outermost layer is made by the non-keratinized stratified squamous epithelium, also known as the corneal epithelium. The cells in the deepest layer of epithelium are columnar. In the middle layers they are polygonal and in the superficial layers they are flattened. So, the cells are arranged regularly. With the electronic microscope we can see that on the superficial surface of the epithelium shows some projections either in the form of microvilli or folds of plasma membrane. These folds play an important role in retaining the film of the fluid over the surface of cornea. So, the corneal epithelium regenerates rapidly after damage. Bowman's membrane. The corneal epithelium rests on the anterior limiting lamina, also known as the Bowman's membrane. Here it is made up of fine collagen fibrils embedded in the matrix. It gives a great stability and strength to the cornea. Corneal stroma. The most of the thickness of the cornea is made up of the substantia propria or the corneal stroma. It is made up of the type 1 collagen fibers embedded in the ground substance containing sulfated glycosaminoglycans. They are arranged with a regularity and form the lamellae. The fibers within one lamellus are parallel to the another. Then because of this regular arrangement of the fibers and because of the fact that the fibers and the ground substance have the same refractive index, the cornea looks transparent. Then the fibroblasts are also appear in the flattened vertical sections but seen as a star shaped from the surface view. They are also called as keratocytes or the corneal corpuscles. Next is the Descemet's membrane. Deep to the substantia propria, there is a thin homogeneous layer called the posterior limiting membrane or the lamina. This is actually the true basement membrane. At the margin of the cornea, it becomes continuous with the fibers of iridocorneal angle. So, the spaces between the fibers of the network are called as the spaces of iridocorneal angle. Then another layer is endothelium. The posterior surface of the cornea is lined by a single layer of flattened cells that constitute the endothelium of the anterior chamber. This layer is in contact with the aqueous humor of the anterior chamber. So, these are adopted for the transportation of the ions. So, they possess numerous mitochondria and they are united to neighboring cells by desmosomes and by occluding junctions. So, these cells pump out the excessive fluid from cornea and thus ensures its transparency. So, now the sclerocorneal junction, that is where the sclera becomes continuous with the cornea. So, that is also known as the limbus. Here the triangular mass of the scleral tissue projects towards the cornea medial to the sinus venosus sclera or the scleral spur. So, in this picture you can observe initially in the corneal epithelium the connective tissues they are arranged regularly, and in the conjunctival epithelium we can make out that connective tissue it is not arranged regularly. Okay, so, the junction between these two represents the limbal epithelium. Now, the histology of retina. The retina is having totally 10 layers. External surface is in contact with the choroid and the internal surface is in contact with the vitreous. So, from external to internal aspect they are having the following layers. The first is pigment cell layer, then layer of rods and cones, the external nuclear layer, external plexiform layer, the internal nuclear layer, internal plexiform layer, the layer of ganglion cells and the layer of optic nerve fibers. Along with these 8 layers, the internal limiting membrane and the external limiting membrane also will be there. So, in the pigment cell layer, it is the outermost layer separated by the choroid by Bruch's membrane. It consists of a single layer of low cuboidal cells containing the melanin pigments. The processes from the pigment cells extend into the next layer. So, that performs the following functions. First is it absorbs the light and also prevents the reflection of light. The pigment cells phagocytize the shed membranous discs of outer segment of the rods and the cones, and it forms the blood retinal barrier between the retina and the choroid. Next is the layer of rods and cones. The rods are the process of the rod cells and the peripheral process is rod shaped. The cones are the process of the cone cells and it is cone shaped. So, in the external nuclear layer we are having the cell bodies and nuclei of rods and cone cells, which are the photoreceptors. They convert the stimulus of the light into nervous impulses. So, the photoreceptors we have seen the rods and the cones. There are about 120 million rods are there and 6 million cones. They are having the outer and inner segment. So, they will be having the photosensitive material, the rhodopsin or the visual purple present in the rods and the hydopsin is present in the cone. Between the second and the third layer, there will be a presence of outer limiting membrane. So, here the zonule adherence of the glial cells with the cell bodies of the photoreceptor cells are present. The external plexiform layer . Here the nerve fibers forms a plexus. The axons of rods and cones synapse here with the dendrites of the bipolar neurons and the horizontal cells. So, the plexiform layer consisting of the nerve fibers, there won't be any cell bodies. Okay. So, the external plexiform layer is made up of the nerve fibers, that is axons of rods and cones with the dendrites of bipolar neurons and the horizontal cells. And this stains lightly. Then internal nuclear layer, it contains the cell bodies and the nuclei of three types of neurons. One is the bipolar cells. It gives off dendrites and that enter the external plexiform layer to synapse with the axons of rods and cone cells. The axons that enter the internal plexiform layer, where they synapse with the dendrites of ganglion cells. So, the bipolar cells are oriented perpendicular to the layers of the retina. So, here we can observe the bipolar cells presenting perpendicularly to the layer of the retina. So, it will be having the two processes, the one is the dendrites and the axons. Dendrites will contribute to the external plexiform layer, then the axons will enter into the internal plexiform layer, and the cell body will be in the internal nuclear layer. So, another type of cell is the horizontal cells. It gives off the process that run parallel to the retinal surface, and these processes enter the outer plexiform layer and synapse with the rods, cones and dendrites of the bipolar cells. The horizontal cells are oriented parallel to the layers of the retina. So, here we can see the horizontal cells, the cell body in the internal nuclear layer, and the two processes are the external plexiform layer. Next type of cell is the amicron cells. It also lies horizontally in the retina. Their processes enter the inner plexiform layer, where they synapse with the axons of bipolar cells and with the dendrite of the ganglion cells. The one more type of cell is present is the cells of the Muller or Muller cells or the gliocytes. These cells gives off numerous protoplasmic processes that extends through almost the entire length of the retina. Externally, it passes to the outer nuclear layer, and at the junction of the layer of rods and cones with the external nuclear layer, it forms the external limiting membrane. Internally, the gliocytes extend to the inner aspect, where they form the internal limiting membrane. So, this membrane separates the retina from the vitreous. The retinal gliocytes are neuroglial in nature. They support the neurons of retina and may ensheath them. So, they will have a nutritive function also. Internal plexiform layer. The internal plexiform layer or the inner synaptic zone consists of the synapsing nerve fibers. So, the axons of bipolar cells synapse with the dendrites of the ganglion cells, and both these processes synapse with the processes of amicron cells. The internal plexiform layer, it also consists of the horizontally placed internal plexiform cells and also few ganglion cells. So, when we observe, we are having the external nuclear layer, external plexiform layer, internal nuclear layer and internal plexiform layer. So, the plexiform layer consists of the nerve fibers. The nuclear layer, there will be the cell bodies and the nucleus. So, next layer is the layer of ganglion cells. It is having the cell bodies of ganglion cells. The axon forms a fiber of the optic nerve. So, each ganglion cells will give off an axon that forms the fiber of the optic nerve. So, next layer is the layer of the optic nerve fibers. The layer of the optic nerve fibers is made up of axons of the ganglion cells. So, the fibers converge on the optic disc where they pass through the foramina of the lamina fibrosa to enter the optic nerve. So, when we observe under the microscope, so we can't make out the structures clearly, but in the nuclear layer as we are observing, we can see the basophilic stained layer. The first layer will be the pigment cell layer, then the rods and the layer of the rods and cones. They are they appears like a strips. Then the next layer, the external nuclear layer, we can see the cell bodies. Then plexiform layer will be lightly stained, that is eosin stained, and the below one will be the ganglionic cell layer, and last is the optic nerve fibers, the layer of the optic nerve fiber. Clinical aspects, the retinal detachment, it is a pathological processes in the vitreous or the anterior segment. There will be a collection of serous fluid in the subretinal space. So, the accumulation of the vitreous under the retina through the hole or tear in the retina. Next is the retinitis pigmentosa. It is a group of systemic and ocular diseases of the unknown etiology characterized by degeneration of the retinal pigment epithelium. The clinical finding is night blindness due to loss of rods. Later it may lead to the total blindness. Retinalblastoma, it is a malignant ocular tumor seen in the children. 60% is sporadic and 40% is familial. Here the child presents with leukocoria, means the white pupillary reflex. Next we will discuss about histology of eyelid. So, the eyelids are a pair of movable the skin folds which protects the cornea from the dust, and it helps in keeping it moist. It is having the different layers. The outer layer is made up of the true skin layer. The skin type is the thin skin, and the hairs are present along the margin of the eyelid as a eyelashes. Below this layer there will be a delicate connective tissue. Then below that there is a fasciculi of orbicularis oculi muscle, and below that muscular layer there will be a mass of fibrous tissue called as the tarsal plate. So, within that there will be a presence of glands which are known as the Meibomian glands or the dorsal glands, and the posterior most it comes in contact with the conjunctival epithelium. So, the tarsal glands will opens into the free margin of the eyelid. The sweat glands, which are the modified sweat glands or the ciliary glands, they are also called as the glands of the mole. They are present near its free edge. The glands of Zeiss means the sebaceous glands in relation to eyelashes, and the glands of Wolfram, these are the accessory lacrimal gland present just above the tarsal plate. So, clinical aspect, style, that is external ardiolum. This is acute suppurative inflammation of the sebaceous glands of Zeiss, apocrine glands of mole, and eyelash follicles. The Shailaja, it is a chronic inflammatory process involving Meibomian glands. It occurs as a result of obstruction to the drainage of secretions. It begins with the destruction of Meibomian glands and the duct and it involves the dorsal plate. Next is the optic nerve. So, the optic nerve is the extension from the brain. So, it is having the meningeal coverings, the outer dura mater, next is arachnoid matter and the pia mater. So, it contains the myelinated axons of ganglion cells of retina. The pia mater sends the connective tissue septa and divides it into a group or the bundles, and they are arranged as the fascicles. So, at the center we can see the central artery, which is a branch from the ophthalmic artery, it is a end artery, and also there will be presence of the central vein of retina. So, under microscope the retina is seen like this. So, we can't differentiate clearly the different layers, but that stain pattern, that is the nuclear cell bodies, they have taken the deep stain, whereas the plexiform layer or the nerve fibers layer, they have taken the stain lightly. So, diagrammatically we can represent the retina showing all the 10 layers. So, cornea, the microscopic appearance. So, here we can make out the non-keratinized epithelium, stratified squamous non-keratinized epithelium, and the posteriorly the simple endothelium, in between these two the stroma. So, diagrammatic representation showing the stratified squamous non-keratinized epithelium and the stroma. Stroma, actually the cornea, it looks transparent because the lamellae and the ground substance are having the same refractive index. Then the optic nerve showing all the 3 layers, that is dura mater, arachnoid mater and the pia mater, and at the center we can see the central vein of retina and the artery. So, the bundles of the axons which are myelinated, they are arranged in the fascicles. So, the questions for assignment: histological features of cornea, histological features of sclerocorneal junction, histological features of eyelid, the histologic features of retina, and the histologic features of optic nerve. So, for the short answers you can draw the only histological diagrams, but short essays you need to explain in detail the microscopic picture of each. Thank you.